and I hope that by the end of this talk, you'll agree with me that the cell surface, while unconventional, is actually a really great place to study nucleic acids. So to begin with, I'm a postdoc in the Flynn lab, and in the Flynn lab, we're very interested in the cell surface. As the first point of contact between the cell and its external environment, the cell surface is particularly important in mediating signals from the extracellular environment into the cell, as well as participating in cell-cell communication. So it's this really important regulatory node for many biological processes. And when we think of the cell surface, we typically think of a diagram that looks like this, where the surface is covered in a number of proteins, lipids, glycans, and various combinations of these biopolymers. But it wasn't until 2021 when my postdoctoral mentor discovered a new type of molecule on the cell surface, and that is a small RNA that is covalently linked to a glycan, and we call these glycoRNA. And these are presented so far on every cell surface from many different organisms that we've looked at so far. Now, as some context and background, I did my PhD studying RNA modifications. So the idea of this novel, brand new, and giant, essentially, RNA modification was very excited to me. And I knew immediately that I wanted to study this for my postdoc. And it turns out that the cell surface is actually full of RNA biology. So some follow-up work from a really talented grad student in the lab, John Purr, showed that the cell surface is also full of RNA binding proteins. And so what he's done here is he's taken live cells, and instead of fixing and permeabilizing them as you normally would for immunofluorescence, he's directly stained live cells with an antibody. So we're actually looking at, in this case, it is DDX21 and DNA PKCS. So these are two well-known RNA binding proteins, and we see they form these really nice clusters localizing to the cell membrane. And so now we're starting to think of the cell surface as something that looks more like this, where there are these nanoscale domains containing RNA binding proteins and glycoRNA, and they form these clusters that are very distinct from other canonical cell surface proteins. So we're really getting these small functional hubs of RNA biology. And so we now have a model that looks like this. And in joining the lab, I was really curious if we have RNA and RNA binding proteins on the cell surface, do they interact? And moreover, could it be that RNA binding proteins themselves are what is tethering glycoRNA to the surface? And so to test this hypothesis, we devised a very simple experiment where we can get rid of all cell surface proteins and look if we can release that cell surface RNA. And so to do that, we just use standard cell culture trypsin. So trypsin is a protease that is used to cleave cell surface RNA to release cells from a plate. And normally with trypsin, you discard the trypsin and harvest your cells. But in this case, I harvested both the trypsin as well as the cell pellet, extracted nucleic acids, and then took them for a variety of downstream applications. So what I'm showing you here is a gel where we've run those nucleic acids, and this is compared to just total RNA. And I've also digested them in vitro with various enzymes to interrogate what these nucleic acids were. And what we can see, very nicely confirming our hypothesis, is we have this lower molecular weight species that we can digest very nicely with RNAs. So this confirms that, yes, we can release RNA from the cell surface when we remove proteins. But perhaps more surprisingly, it was this higher molecular weight band that was resistant to RNAs, but disappeared completely with DNAs. So once I got over the shock of having to pivot from RNA biology to DNA biology, I decided to dive further into this. And the next thing that I did was to sequence this material. So I sequenced that trypsin DNA as well as whole DNA from the cells. And if we align it to the nuclear genome, we don't see any major enrichment across any of the chromosomes. But all of a sudden, if we map these reads to the mitochondrial genome, we see this really nice enrichment of mitochondrial reads specifically from the trypsin DNA, which confirms that we are able to release not only DNA, but mitochondrial DNA from the cell surface. But this wasn't quite the evidence that we were hoping, since we were just able to show that we could release DNA, but not actually that DNA was present, stably tethered to the cell surface. So we sought to develop an assay that could confirm and, in fact, visualize this cell surface DNA. And we took inspiration from ATAC-C or ATAC-seq. You might be familiar with this method for looking at chromatin accessibility. The way this works, you start with your cells and you fix and permeabilize them. You then add an enzyme called a transposase, or TN5, that you load up with oligos. And you can customize these oligos however you wish. You can add fluorophores so that you can visualize these by microscopy. You can add a biotin, you can add handles for sequencing. And the idea is that when this transposase, complex with oligos, can find accessible regions of double-stranded DNA, it will integrate these oligos into the DNA. And you can then infer regions of open or closed chromatin based on the frequency where you get these transposition reactions. So in looking at this, we reasoned that if we omit this initial fixing and permeabilizing step, we can instead add the transposase directly to live cells. And if there was DNA on the surface, we would be able to visualize this. And so that's what I'm showing you here right now. These are leukemic cancer cells, AML3s. At the bottom, this is where we've added our fluorescently labeled oligos, but no enzymes. That's this minus TN5. And at the top here, this is where we've added the fluorescent oligos as well as the enzyme. And we have a 3D representation here where we can see that we have these really nice bright puncta that localize to the cell surface and kind of surround the cell. So they really are localizing to that membrane. This was all very exciting to us, but again, we wanted to confirm that this was actually on the cell surface. So we took cells and subjected them to various live cell enzyme treatments. So we can see these nice bright spots when we don't do any enzyme treatments. But when we treat cells with benzonase, so this is an all-purpose nuclease digesting both DNA and RNA, we almost completely lose our cell surface signal. But importantly, this is not the case when we treat cells with RNAs. So this goes to show that not only is this DNA accessible to live cell enzymes, but it really is DNA, and it's not our cell surface RNA that we've previously characterized. And so taken together, this method, which we call cell surface ATAC-C, or CS ATAC-C, allows for really rapid in situ characterization of cell surface DNA. And I want to emphasize that it really is rapid. This entire reaction only takes 5 minutes, and we hope that the ease and speed of this reaction will allow others to adopt this protocol and start looking at DNA on various different tissue and cell types. And so having this method in place, we could now start getting into really exciting microscopy applications. So the first thing we did was team up with the MoCo lab at Max Planck to do some super resolution microscopy of our cell surface DNA. So what I'm showing you here, again on cancer cells, in blue, we have our TN5, so this is the DNA. And in pink, I'm showing you beta-2 microglobulin, which is a classic cell surface protein, and this is just meant to localize and orient you so you can see the cell surface. And what we can see is that although these spots are quite few in number, they really are quite large. They measure to about a micron or more, which is quite big when we're thinking of cell surface molecules, and they are, of course, very bright. This is in contrast to the RNA binding protein clusters that we've previously seen, where those are a lot smaller, so they're typically around 100 nanometers and form this kind of tessellated pattern on the cell surface. So they're both highly abundant and small. Meanwhile, our DNA is less abundant in terms of cluster number, but very large. So it seems that these two clusters do seem to be biophysically quite distinct. Now, with all of this, we really wanted to be certain that not only were we looking at DNA on the cell surface, but that this DNA was on the external side of an intact plasma membrane. So it wasn't just that the plasma membrane would rupture, release all of its contents, and we would see DNA on the surface. And so to do that, we took advantage of scanning electron microscopy. And the way this works is you have your sample oriented at an angle relative to the detector, which allows you to look at the topography of the cell surface. And so just showing you some previously published examples, this is looking at cancer cells, and we get these kind of funny looking images where these cells are covered in these strange protrusions. This is normal. These are when glycans on the cell surface can shape the membrane and push it up into these protrusions. So we see regions of the cell surface that are a bit smoother, as well as these kind of rougher regions. So here with our DNA, we could swap out that fluorescent oligo with one containing biotin, which would allow us to come in with a streptavidin gold and perform this scanning electron microscopy. And we did this in collaboration with some really talented people in the Harvard EM core. And so what we can see here, I'm showing you two different regions of the cell surface, this smoother region, as well as the region with these glycan-mediated membrane protrusions. At the top, we have our no enzyme condition, and at the bottom, this is where we have our enzyme as well as our oligos. And what we can see are these really high contrast spots, which represent the cell surface DNA. And we can see several things. Number one, it is definitely located on the external side of the plasma membrane. And we can see in both of these cell surface topologies that it really is an intact plasma membrane. So it's not that the plasma membrane is breaking. And finally, if we look at the approximate size of this cluster, it really is in agreement with our super resolution measurements, showing that these clusters are about 1 to 2 microns in size. So it's a really nice orthogonal measurement of the structure of these molecules. So moving forward, having started to convince ourselves that DNA may actually be a novel component on the cell surface, we wanted to investigate this further by looking at how the DNA was organized relative to other molecules on the cell surface. And so again, we used our biotinylated probe, which would allow us to come in with a streptavidin HRP and biotinylate regions around the DNA on the cell surface so that we can get a proximal proteome. And so these are some of the hits that we've gotten, which you might recognize, both because one of the first things that came up was cell surface proteins. This was nice confirmation for us that we really are looking at topological specificity. So a lot of what we're looking at is limited to the cell surface, and we're not getting internalization of the labeling reaction. But perhaps more excitingly to me was that one of our top hits was RNA binding. So a lot of these proteins like nucleolin, DDX21, and nucleophosmin are the classic RNA binding proteins that we see on the cell surface. So luckily, I haven't strayed too far from the RNA yet. We wanted to confirm these findings by confocal microscopy. So I co-labeled cells with the TN5 to look at DNA, as well as 2 common reagents that we use in the lab to look at cell surface RNA biology. The first is this protein, this recombinant Siglec-11. We know Siglec-11 is a really strong and specific binder of glycoRNA, so we can use this as a proxy for looking at glycoRNA. And this NPM1, which is a cell surface RNA binding protein. And so what I'm showing you here is that we see this really nice co-localization between the DNA and these RNA regions, which I've quantified here in this graph. So this graph is looking at the distance between 2 points that we're seeing in the microscopy image. So on the x-axis, we have the distance between 2 points. And on the y-axis, we have the fraction of pairs that are at that density. So when we see something that's higher up on the y-axis, but close to 0 on the x, it means we have a high degree of co-localization, which we can see between the DNA and Siglec-11 and nucleophosmin, but we don't see that same degree of co-localization with standard cell surface proteins, such as the glycoprotein CD44 or an integrin. So it really confirms that even though our DNA clusters are a lot less numerous than the RNA and RBP clusters, they really seem to be sharing the same neighborhood. So it is exciting that we do have these links between DNA biology and RNA biology on the cell surface. But finally, the question that everyone was asking us, that we were asking ourselves, was whether or not cell surface DNA can play a role in disease. And so the first thing we wanted to do was see if we could detect surface DNA on primary human cells. So all of the data that I've shown you up until now has been on cancer cell lines that are grown in culture, but we wanted to see if we could extend this and move in vivo. In order to do that, we had to adapt cell surface ATAC-C a little bit to see if it was compatible with flow cytometry, and thankfully it was. So with the exact same conditions that we can use to label cells for confocal microscopy, we can also do flow cytometry. And so I'm going to show you a lot of graphs that look like this, where the gray histogram is where we have our no TN5 reaction, and in orange, this is where we've added TN5 and the oligos. And we see this really high signal over the no enzyme control. This is, again, just looking at cancer cell lines. It's nicely sensitive to enzyme activity. So we've already known from our confocal data that enzyme activity decreases when we incubate cells on ice, and we can recapitulate that nicely here. And another neat thing that we can do with flow cytometry is look at whether cells are dead or dying, because another one of the questions that we were wondering was whether this was just a function of dead cells releasing their DNA. And so what we've done here is we've done our DNA staining and combined that with annexin-5 staining. Annexin-5 is a protein that binds to the surface of apoptotic cells, so we could discriminate live cells, dead cells, and cells that are in the process of dying. And if we look at these annexin-5 negative cells, so these are our live cells, we still have that really high amount of cell surface DNA, suggesting that not only is cell surface DNA present on live cells, but it's also not a function of cells undergoing apoptosis. So with that in mind, we collected samples. So these are primary human peripheral blood mononuclear cells on the left and bone marrow mononuclear cells on the right. And we performed our CS attack-seq. And the first exciting thing that we found was that there's this really clear cell type specificity. So if we look at B cells or monocytes in both sample types, we see this really nice signal over our background that we don't see when we look at T cells or NK-like cells. So it really does seem that cell surface DNA is not a universal thing that's found on all cell types, but it's really restricted to one or two cell types, which really lends precedence to the idea that it might have a function. And so we asked whether the cell surface DNA was relevant in disease. And serendipitously, we came across this paper talking about an extracellular nuclease called DNase 1L3. So different cell types in your body secrete nucleases, so DNases and RNases. You might be familiar with DNase 1. This is kind of your classic DNase, similar to what you would use in a lab, and is really good at digesting naked DNA. But there's also a DNase paralog called DNase 1L3, which is really good at digesting DNA that is complexed with proteins or lipids. And it has mostly been characterized for its role in digesting DNA on microparticles. So microparticles are these membrane-encapsulated vesicles that are released from apoptotic cells and tend to present DNA on their cell surface. And while DNase 1 is not able to access that, DNase 1L3 is really great at digesting that microparticle DNA. And what's important is that in mice where you have deleted DNase 1L3, these mice tend to develop antibodies against double-stranded DNA. And in the clinic, antibodies against double-stranded DNA is a hallmark of autoimmune diseases such as lupus, although we don't know why exactly that is. But this did seem to suggest that there was some relationship between extracellular DNA and autoimmunity. And sure enough, this putative link was further explored in the context of a cohort of patients with systemic lupus erythematosus that all had a loss of function variant in DNase 1L3, again suggesting that there's some unknown link between this enzyme and the ability to produce these antibodies against DNA, which eventually leads to autoimmunity. And so we teamed up with the lab of Kim Simpfendorfer, who is an expert at DNase 1L3. And the very first thing we did was treat our PBMCs with recombinant DNase 1L3. And so what I'm showing you here in black, this is where our no enzyme treatment. So it's just our cells that haven't been treated with any DNase. And in blue, this is where we've treated our cells with DNase 1L3. And what we can see very clearly is that in the cells, particularly the B cells and the monocytes that are treated with DNase 1L3, all of a sudden their cell surface DNA decreases. So DNase 1L3, not only is it capable of digesting that microparticle DNA, but it is also able to digest DNA that is on the cell surface. And so we went further into looking at B cells. We isolated B cells. And what I'm showing you here are actually my own B cells, which is very exciting to see that they do have DNA. And what we can see is that when we treat them with DNase 1L3, again, we can remove that cell surface DNA, but it's very specific to DNase 1L3 because when we treat cells with recombinant DNase, we are not able to digest it. So there really is something special about DNase 1L3 that gives it this cell surface function. But with all of this, we were exogenously adding enzymes, which didn't necessarily convince us that DNase 1L3 had a role in vivo at regulating cell surface DNA. And so again, with the help of Kim's lab, we obtained two sets of patient samples. In black, we have our patients with the wild-type allele of DNase 1L3. And in green, certain patients have a missense mutation in DNase 1L3 that impairs its secretion. So these patients are still able to make the enzyme, but the enzyme is not able to be secreted into the extracellular environment to digest DNA. And very gratifyingly to us, we saw that these patients without functional secreted DNase 1L3 suddenly accumulated really high levels of cell surface DNA on their B cells, more than what we would see in healthy patients. And even more surprising, they even started to accumulate DNA on their T cells, which normally did not have DNA. And what's important to note about this patient variant is that although these patients are currently healthy, this risk allele is associated with increased development of autoimmune diseases like lupus, which again really suggests that there's this intimate link between the ability to clear DNA, either in the extracellular environment or on the cell surface, and the susceptibility to autoimmune diseases. So now I'm also going to show you some data that is still in progress, but we wanted to look at actual patient cells. So with a collaboration with Morgane and George, also at Boston Children's Hospital, we gained some PBMCs from patients that actively have lupus. And it's worth noting that all of these patients do have antibodies against double-stranded DNA. And this is still a work in progress, so we've only looked at a very small set of patient samples, but we can start to see this dysregulation of DNA, particularly on monocytes, where these wild-type patients, again in black, and the autoimmune patients in pink, they start to accumulate higher levels of DNA. And again, some of these patients also start to accumulate DNA on their T cells, which really suggests that being able to regulate cell surface DNA may be an indicator of susceptibility to autoimmune disease. And so to sum up what I've told you a bit about today, I've shown you that mitochondrial DNA is a new component on the cell surface that we're only just beginning to characterize. We've also established a method called cell surface attack, which allows for multi-omics spatial information. So we can sequence the DNA, we can look at the proximal proteome, and we can also visualize it by various different types of microscopy. And finally, this protein DNase 1L3 has a role in maintaining levels of DNA on the cell surface, and particularly in keeping these levels to a certain degree so that they don't overwhelm the surface, which may lead to autoimmune disease. Now, before I go and start thanking all of the people who are involved in this project, I wanted to spend the next few minutes of my presentation talking about something else. So as you may have guessed, although I've spent a lot of time talking about DNA, I'm actually very excited about RNA. And in particular, one of the reasons that I was really looking forward to presenting in this seminar series was that many other postdocs and PIs who've presented have taken the time to present their own initiatives related to EDI. So I really wanted to discuss with you a topic that I am passionate about. And I am part of RNA Canada, which is a community of RNA researchers all across Canada. And in particular, I'm part of the EDI committee. So I get to work with this really fantastic group of people led by Michelle Scott at Sherbrooke University. And so I wanted to share with you some of the things that we've been working on. And I would encourage you to go to our website here at the bottom. Under the About Us tab, you'll find a tab for EDI. And so one of the things that we've done is provide some EDI resources for researchers across Canada. So we have in English and French our EDI policy, and we also have some guidelines for conference organization. These guidelines are not meant to be prescriptive, but we hope that if you are organizing a conference, you'll be able to take a look at these and implement some of these into your own organization and planning. We also have a list of EDI resources, and this is constantly being updated, so keep your eye out on it to see more. A lot of these, we were also fortunate enough to work with Dr. Imogen Ko, who is a regular attendee of these CSMB presentations, and she listed some of her favorite EDI resources, which I am now sharing here. And finally, we also host workshops, both in person at various RNA meetings across Canada, as well as online. So in 2024, again, we worked with Dr. Imogen Ko to deliver a workshop at our annual conference. And what I'm just showing you here is a summary of what conference attendees thought EDI means after they attended the workshop that Dr. Ko presented. And on our website, you can actually access all of this information. So we have the slides that Dr. Ko presented. We have some interactive activities for looking at scenarios, and you can use them to discuss with your lab members what kind of EDI issues are there and how you might resolve them. And finally, I'm very excited because we have an upcoming online EDI workshop, which we're presenting working together with the Toronto Initiative for Diversity and Excellence. And so in this workshop, we'll touch on various things that affect both trainees and PIs as they relate to EDI. So things like how to incorporate EDI into scholarship applications and how to establish an inclusive environment in the lab. Registration will open soon, and it is free for RNA Canada members. So I would really encourage you to take a look at RNA Canada, whether you're new to RNA or you've been studying this for a while, and consider joining us. And with that, we have very many people to thank, my entire lab, including my supervisor, Ryan Flynn, many collaborators. We have Kim's lab, who helped us with all of the DNase 1L3 work, Ivan and Valentina, who did some of the mouse work that I didn't have a chance to talk to you about, Leonhard Mokel's lab, who helped with the super resolution microscopy, the Z and Garcia labs, who did a lot of our mass spec, Sunit Agarwal's lab, who helped with all of the mitochondrial aspects of this, and finally, George and Morgaine, who provided us with patient samples. And with that, I'd be happy to take any questions.
Much, Dr. Jennifer Porat, for this great presentation. So please, everyone, feel free to raise your hand and give your question. If you're feeling shy, you can also write your questions in the chat, but we prefer to have it all live. I'll do a quick start while you guys are thinking. It's already my third coffee, so I'm survived the shock that DNA now is on our cell surface as an integral part of it. And
Yep.
my question is related to more like you must be speculating what's the point of it, right? Because the way I see it, okay, it needs to be digested to prevent autoimmune diseases.
Mhm.
But do you think it is there in the first place to help the immune system because it's mitochondrial DNA? So my first thought was, okay, they put it out there to really ensure that they don't attack their own DNA, which is like my own body's DNA most likely to be from a pathogen would be mitochondrial, right? Or do you think
Exactly.
these clusters have any other purpose?
Yeah, that's a great question. Short answer, we don't know yet, but are in the process of figuring out. A longer answer, we think yes, it is most likely involved in immune responses. So the fact that this is mitochondrial DNA, which really resembles bacterial DNA, raises the question of how this might be involved in self versus non-self, pathogen detection. And what we think is that maybe the DNA that's on the surface is kind of dampening the initial immune response so that if pathogen DNA does come in, the cell is able to somehow integrate these signals. And we're not quite sure how that works yet. Another possible explanation is that it's not necessarily the DNA itself that's important, but really the negative charge. So we know from the RNA clusters on the cell surface that they form these dense regions of negative charge that are really important for binding and internalizing growth factors and cell penetrating peptides. And so again, this is not necessarily the nucleic acid itself that's mediating this, but rather just having that localized region of charge. But of course, that begs the question, if any nucleic acid could be there, why would you specifically have some cells that have DNA, some cells that have RNA? And so we're really curious about that and looking into these questions.
Okay, thank you for answering the question and shattering my basic knowledge in science. If I heard about it yesterday, I would have said, no way, there's double-stranded DNA on our cell surfaces, but apparently it is. All right, do we have any questions in the chat or anyone bold enough to raise their hand to ask a question? I see one. Mark, stage is yours.
Well, I'm worried that this is meant to be for trainees, so I don't want to
Ask away.
take this space at the so it okay, I'll ask. Do I understand correctly? It's a great talk. So cool.
Thank you.
Do I understand correctly that you have fewer of these sites than you have that you have RNA associated sites,
Yes.
and in the sites where they exist, there are many. They're kind of there are puncta within the sites, and so I guess the question is, what else is specifically in those sites that might explain why it's not more broadly bound around the cell, I guess, something like that.
Yeah, exactly. So we know that the RNA is, they're very small sites, but many of them, the
Yeah.
DNA, to be that a lot of DNA clusters in that one particular site. As for what else is there, we know that there, so some data that I haven't shown here, we know that there are proteoglycans like heparin sulfate proteins that help nucleate and anchor these DNA clusters. And we also know from our proximity labeling that not only are there a lot of RNA binding proteins, but the RNA binding proteins that we do find, like DNA PK and nucleophosmin, they've also been reported to do double duty as DNA binding proteins.
Okay.
We haven't formally shown a direct interaction between the DNA and these proteins, but it stands to reason that if they are together, if they're proximal, they might be interacting. And so it seems, again, similar to the RNA foci on the cell surface, there are really these small regions that are dedicated to nucleic acid biology.
And quickly, if I can, if the idea is that, if one of the things that's on your mind is that, what's the purpose of this, is it worthwhile looking in kind of going down evolutionarily? Like, do you see it in Drosophila? Do you see it in C. elegans? Do you see it in, you know what I mean? And then based on when it starts to emerge, you might start making guesses as to like, okay, what's
What's
its
it
function?
doing? I would
Yeah.
love to do that. So for now, we've only looked at humans and mice. So it does seem highly conserved in mice that we only see it on B cells and monocytes. As of now, that's all that we've done because we don't have access to the same model organisms that I would like to. But I think in the future, I would really like to look at how widespread this is. And then once we can establish that, again, as you said, if it emerged at this particular point, what other traits co-evolved with that might help
Sure.
us determine function.
Thank you.
Okay, if there's no further questions, don't worry. You all get a second chance because now we can go to our second talk and I'll give the stage to Dr. Elina Karimullina. Stage is yours.
All right. Thank you so much for having me here today. First of all, I'd like to start with, how should I? I'm sorry, I just need first to organize my screen. So first of all, I'd like to start with land acknowledgement. University of Calgary, located in the heart of southern Alberta, both acknowledges and pays tribute to the traditional territories of the peoples of Treaty 7, which include the Blackfoot Confederacy, comprised of the Siksika, Piikani, and the Kainai First Nations, the Tsuut'ina First Nation, and the Stoney Nakoda, including Chiniki, Bearspaw, and Good Stoney First Nations. The city of Calgary is also home to the Métis Nation within Alberta, including Nose Hill Métis District 5 and Elbow Métis District 6. And I hope to see you all in Banff this spring because I'm definitely participating in that conference. So today, I'll show how we combine sequence and predicted structure guided annotation, and then take targets to the wet lab for expression, purification, structural validation, and functional assays. But first, why Pseudemonas aeruginosa? Pseudemonas aeruginosa is an adaptable, opportunistic gram-negative bacterium that thrives in many environments, from soil and freshwater surfaces to host-associated niches. What makes it clinically challenging is its ability to persist. It combines intrinsic and acquired antibiotic resistance, strong biofilm formation, and a capacity to cause chronic, difficult-to-clear infections across many hosts, including humans. The image here shows a natural Pseudemonas aeruginosa biofilm just on a granite pebble. And in the higher magnification view, you can see the rod-shaped cells embedded in the extracellular matrix, which is a key contributor to protection and persistence. This biofilm lifestyle isn't just a phenotype, it fundamentally changes envelope biology, and the outer membrane becomes a major determinant of what the bacterium can import, export, and resist. And the clinical footprint is broad. It's not a single niche pathogen. This figure from the recent review paper summarizes the clinical breadth, respiratory disease, wounds, urinary tract, bloodstream. Across these settings, the recurring theme is surface biology. The outer membrane is the interface for nutrient acquisition, immune interaction, and antibiotic permeability. And here is the paradox that motivated this project 3 years ago. For something so central, a large fraction of outer membrane proteins remained poorly annotated or simply called hypothetical. So we asked the practical question, what do we actually know about the outer membrane proteome when we annotate it rigorously? This figure makes a key point. What you find depends on how you look. Using close homology searches, you recover the obvious domains. With remote homology, you recover more. And with structure-based comparisons of predicted models, you reveal additional domains, especially in proteins that are modular or diverged. In other words, the outer membrane proteome isn't just a list of barrels. It's a set of barrel-centered machines, often with functional domains fused on. And once we saw that, we stopped treating these as porins in the generic sense and classified them by architecture. This breakdown shows the major beta barrel categories we recover using evolutionary classification of protein domains database. Porins, ligand-gated or TONB-dependent channels, plus smaller groups like OMPA-like autotransporters and transport proteins. But beyond the main barrel families, the real hidden complexity is in the extra domains. This is the part that makes this data set biologically useful. The additional domains, like TONB-dependent plaque, POTRA domain, and others, appear in non-random combinations. For example, ligand-gated beta barrel often come with plaque domains, and other architectures frequently pair with periplasmic domains that likely tune assembly, stability, or interactions. And once you see the codomain patterning, hypothetical outer membrane protein stops being a dead end. It becomes a testable mechanistic hypothesis. And we didn't just stop at the annotation. Next, we asked, can we produce real proteins out of these annotated sequences in the wet lab? And this slide is our prediction to bench proof of work, a clear funnel. We started with 105 annotated Pseudemonas aeruginosa outer membrane beta barrel candidates. Then we prioritized 60 for structural work and moved them into experimental validation. 18 targets expressed and could be extracted from the outer membrane, and 17 were purified at a large scale. So far, we solved 3 structures at high resolution and with more currently in progress. And next, I'll show 2 structural case studies and 1 functional example where prediction and experiment provide complementary insights. One of experimentally solved structures is EPRS, a subtilisin-like serine protease autotransporter. EPRS can activate protease-activated receptors on human epithelial cells. It does this by cleaving their tethered ligand regions, and this triggers inflammatory signaling pathway in mammalian cells. We resolved the passenger domain at 2.34 angstrom and the beta barrel translocator at 3 angstroms. And we then combined them into the first full-length structural model. And the structure shows a continuous autotransporter, classical autotransporter architecture. A beta barrel and a linker lead to a beta helix talk and an amino terminal globular region. Structural comparison supports a canonical subtilisin-like mechanism. The model positions the catalytic triad, and it also highlights the conserved motifs near the cliff that likely stabilize the reaction intermediate and help orient the substrate, since it's a 2-step mechanism of action. Next, we validate predicted structure. And we used the PRS to evaluate AlphaFold3 prediction quality. And the global fold is captured well, including the catalytic core. But the difference is cluster where we expect physics to matter most: boundary regions, loops, and solvent exposed strand edges. The predicted model tends to over-regularize beta structure. It makes loops look like strands and makes flexible regions look tidy, while cryo-electron microscopy shows that those regions are often flexible. And we also tested outer membrane protein from another outer membrane protein family for the same pattern. Its PA2590 protein is particularly interesting because it's been annotated as hypothetical in major resources, and yet our structure-based annotation places it as a TONB-dependent outer membrane receptor. We determined the cryo structure, and the protein adopts the canonical TONB-dependent architecture. It's a 22-stranded beta barrel in yellow with an internal plug domain in red, occluding the lumen. And the extracellular loops form the entry surface and shape the binding pocket. This is the extracellular site. This is the periplasmic site of bacterial, and this is the just schematic representation of outer membrane. Recently, there has been a paper released showing that PA2590 is a virulence factor in eye infection, and some docking data with heme and vitamin B12 with predicted structure were done, but ligand for this transporter is still unknown, and we're also working on that. We did the same analysis, aligning experimental structure with predicted, and we observed the same pattern of deviations in extracellular loops and periplasmic turns. The predicted model renders those regions as well-ordered secondary structure, including beta strand extensions and even small beta sheets in loop regions that appear disordered experimentally. So the same message, prediction seems to be good for fold assignment, but if you care about gating specificity, antigenicity, permeability, the loop ensemble is where you need experimental data still. And some more of no unknowns, the protein PA4579, the one that is annotated as hypothetical protein, and even the localization of this protein is not annotated, certainly for outer membrane in Pseudemonas.com database. But we annotated this target as outer transporter since it contains a 12-stranded beta barrel fused to domain of unknown function for 105. And structurally, that DAF domain aligns with LNB, a lipoprotein N-acetyltransferase from Bacteroides. And the structural similarity is strong here. RMSD is 1.7 angstrom, and importantly, the predicted active site residues align in a way that suggests a conserved catalytic architecture. And this gave us a hypothesis that PA4579 may have N-acetyltransferase-like chemistry, despite being in a very different organismal context. So we set up a biochemical assay to test whether PA4579 can acylate a substrate. And we used FSL1 biotin as an acceptor substrate and phospholipids as donors. FSL1 biotin is detected by streptavidin horseradish peroxidase, and an acylation-dependent mobility shift reports activity. In our preliminary experiments, PA4579 shows acetyltransferase-like activity, but these data are not even half-baked. They are quite preliminary, and we are now tightening the mechanism with controls, including a catalytic cysteine mutant, to confirm that the activity tracks with the predicted catalytic site. So stepping back, what did we learn across the whole project? An integrated workflow identifies 105 candidates, minder beta barrel outer membrane proteins. And these ultramembrane proteins partition into major classes, and their codomain pairing is non-random, which gives functional priors for uncharacterized targets. Crucially, this goes prediction to bench. We expressed and purified a panel of ultramembrane protein targets, including multiple hypothetical proteins, enable downstream characterization. Across targets, AlphaFold captures global fold well, but tends to over-regularize beta-strand continuity, exactly where cryo-EM often reveals flexibility. And PA4579 is an example of function discovery. Structural annotation led to a catalytic hypothesis, and preliminary assays indicate acyltransferase-like activity. The impact is that we now have a roadmap that scales computational detection, expression, and scale up, structural validation, and functional assays. And it produces a prioritized target set for substrate discovery, inhibitor design, and antigen selection. And next, we're expanding expression and biophysics and cryo-EM studies to additional outliers and newly expanded families. The goal is to keep converting unknown outer membrane proteins into mechanistic, testable biology. Big thanks to the team and collaborators listed here and to everyone in Savchenko laboratory. This work is deeply collaborative across computation, cloning, biochemistry, and cryo-EM. Thank you, and I'd be happy to take questions. Hello.
Thank you so much for your presentation. Now a new chance. Are there any questions in the chat or any raised hands? Oh, we got a hand. Where we are. Oh,
Yes.
was that him?
Really great talk.
Go.
I was
Thank you so
wondering, when you express and purify these proteins, do you do it with the membrane? And if not, would you expect that plus or minus the membrane, it would adopt different conformations?
So for membrane protein, integral membrane proteins, the major thing is to solubilize membrane. So there is a specific step of extracting bacterial membrane. And then we test, within the last 5 years, there have been so many breakthroughs in organic chemistry of compounds that actually can pretty well stabilize membrane proteins in solution. And that's what we use. Whenever we use detergents, we fail with cryo-EM single particle reconstruction, for example. But as soon as we reconstitute in nanodiscs, amphipoles, SMALPs, yeah, it's a completely different game there. So yeah, that's our approach. You need to extract membrane, then you need to solubilize it, and then you need to stabilize your membrane protein in solution. And then you can work with that, whichever it's a functional study or structural study. Yeah, that's how it works.
That's very cool. Thank you.
Any other?
Questions? Yeah, we got one more. Stage is yours.
This comes from a place of very low expertise, so I'm apologizing if it's not.
Oh, it's the same. It's the same from RNA to protein. Yes, the focus is completely different. I agree. Yeah, no worries.
But it strikes me as kind of this awesome pipeline you have for characterizing all these new proteins with different folds and membranes is not something that's unique to Pseudemonas. It can be done for all sorts of things, eukaryotic
Of
cells.
course. Of
So
course, yes.
is there a reason that you've kind of, I mean, obviously there's a virulence narrative, which is great, but I mean, or is it something that you're kind of bringing to other kinds of membrane questions, or is it other bacteria? You tend to keep to bacteria, or is it
Yes,
because it
yes.
sounds like this is just, you
This is
know,
the
what
approach.
you're
This
doing
is
with the
the
cryo-EM. It just sounds like it's the beginning of something that can just go in many directions in a big way.
Yes, that's true. So first, I would I probably should start with the approach that usually scientists, what people usually do when I attend the membrane protein conferences. Pretty often, PIs tell the story that they've been working only on one protein for 20 years, and then they stop because it was so hard to work with that. But there is another approach, and that's approach in Alexei Savchenko laboratory. First, we try to come up with a huge list of membrane protein targets to work with. And we search homologs as well. And we are not focused only on one. This project is just 1 out of 5 that I have on my plate right now. And the approach is pretty much the same. Only since I'm a research associate in the Department of Microbiology, Immunology, and Infectious Diseases, our focus is on escape pathogens. Escape pathogens are those nasty bacteria, Pseudemonas, Klebsiella, Enterobacters, and yeah, so we use it, and it's quite successful because, as you could see, out of 60, only 18, only 1/3 was expressed, and only 1/3 was we were able to solubilize it and to do downstream production, downstream biochemistry with those proteins. So yes, this approach. There is a paper on Legionella, and I think it's the first. It's called Atlas of Legionella effectors. And we sometimes make fun of the word atlas because it can be applicable to any our further work, because we do this kind of atlas for different pathogens. Yeah. Yeah.
Your answer makes me think of a conversation I had with another structural biologist who told me that he only reads the papers about the thing that he's working on after they get good structures.
Yes.
They cast a very wide net, and then the things that they get, then they become experts about it. Instead of
Yeah.
what you
Yeah.
did, which is, you work on something for one thing for 20 years, and then you decide, okay, no, it's not going to work.
Yes, it's true. It's true. And it's amazing how within 5 years, the field changes so fast with cryo-EM. It's amazing. Like 5 years ago, I was just taking the sequence and searching for papers, and there was practically nothing. And then with cryo-EM, like comparing what you were thinking about this protein and what you've got, it's such a huge difference. Yes, and only after you get the structure, you start mining on yeah, on its characterization and how it works, and yeah.
Thank
It's a
you.
very fast, yeah, delivering field. No, thank you. My pleasure.
All right. If there's no further questions, I would like you all to join me in thanking our two speakers one more time for those excellent talks. And I hope